Okay, it's my great pleasure to be here and present our work, the GWAS of PTSD among childhood cancer survivors. My name is Donghao Lu. I'm an epidemiologist and assistant professor at Karolinska Institute in Sweden. First of all, I have no financial disclosures or conflicts of interest. So it has been well documented that childhood cancer survivors are at risk of PTSD. And in the previous reports, that has been shown up to 20% of the survivors could have significant symptoms, PTSD symptoms years after cancer treatment. And it has been several risk factors have been identified, including brain radiation, intensive cancer treatment. And also we know that females and survivors with lower socioeconomic status are more often to report PTSD symptoms. However, we know very little that why some survivors go on and develop PTSD symptoms while others doing pretty okay. One of the theories is that genetic predisposition might shape the PTSD development among the survivors. And we do have a lot of evidence now from the other or general populations showing that we're demonstrating the genetic contribution to PTSD. And twin studies have shown that the heritability of PTSD is up to 71%. And the very latest GWAS studies with a very significant number of cases, like more than in total, more than 70,000 cases have identified 6 risk loci. They all came out during the past like 3 years or 2 years, like the Psychiatric Genomic Consortium with 30,000 cases, they have identified 2 risk loci for PTSD. And the Million Veteran Program have identified 3 risk loci, also like 30,000 cases. And ISAC is a Danish biobank, and they have found 1 risk loci for stress-related disorders, including PTSD. However, those GWAS, for example, like PGC and MVP, they're dominated by the veteran populations. It is unclear whether those findings could be generalized to the cancer populations. And also often the controls in those GWAS may not have experienced any traumatic events. If they do, maybe some of them will develop PTSD symptoms. So those could lead to attenuated associations in the findings. Although I know many of you must have heard or known the Genome-wide association studies, but just a little bit overview of this kind of GWAS that we use. We often rely on the large cohorts or case control studies. We collected the DNA samples for genotyping, and after quality control, and imputation, we will get like millions of the single nucleotide polymorphisms called SNPs for each individuals, and then we can use registry regression, linear regression to analyze the association between the SNPs and the outcome of interest. And then we can put into a Manhattan plot showing here that if we found a very significant marker of the genetic variance, then we'll top up on this figure with a very small p-value. And often we will need to do meta-analysis, meaning that we need to combine different cohorts for power sufficient analysis. And we often need replication in an external cohort or study to sort of confirm these findings. And it's very useful to do the post-GWAS analysis to sort of functional annotation to provide insight on the potential biological mechanisms afterwards. So in this study, we conducted a GWAS to characterize the genetic signatures of PTSD in the childhood cancer survivors. And we also illustrated the potentially shared and or different genetic architectures with PTSD following other traumatic events. So our study population is based on the CCSS, which is our discover setting. And CCSS is a retrospective large cohort with 31 collaborating centers in USA and Canada, included the major pediatric cancer patients diagnosed before age 21, and they have survived over 5 years under recruitment. And in the original CCSS, there are like 14,000 participants being enrolled, and about 41% of them have available genotype data. And because we need those participants have completed the PTSD measurements, so we are down to 3,984 participants of European ancestry in analysis. And we used the Sanju lifetime study for the replication. Sanju Life is also a big cohort, included all pediatric cancer diagnosed in Hsinchu Hospital, and those participants also survived over 5 years in the recruitment. And there were 1,467 participants of genotype data available and of European ancestry. So in the CCSS cohort, the PTSD was measured using the post-traumatic stress diagnostic scale called PDS. And this scale was sort of like revised to measure, like specifically to measure the experiences against the cancer, the childhood cancer, not other kind of trauma. And this was done in the follow-up questionnaire in 2003. That was like 24 years averagely after the cancer diagnosis or cancer treatment. And we classify the PTSD cases based on the DSM-4 criteria, which has been established in the previous study. And that is including if someone reporting one or more items if they are re-experiences symptoms, 3 and more items of avoidance symptoms, and 2 or more of arousal symptoms. We also studied the PTSD symptoms as continuous variable. We calculated the total score of the symptoms, and also we estimated the subscores like re-experiencing score, avoidance score, and arousal score using the corresponding symptom items. And in Sanju Life, this was PTSD was assessed using PCLC questionnaire, but this is not specific to cancer. Could also be other kind of traumatic events in their life. And we classify the PTSD cases using the well-established cutoff for the Sanju Life participants. And regarding the genotyping, the CCS in CCSS, we have the GWAS sort of genotype data, and we did imputation so that we have 20 million sort of SNPs for analysis. In Sanju Life, that was the whole genome sequencing. So in the CCSS cohort, we identified 671 PTSD cases, which is like 17% of the participants. And the mean age and cancer diagnosis was 8, and the PTSD assessment was like 32 years old when they were. And we have seen that PTSD cases are more likely to be a CNS tumor, and also received the radiotherapy back then. And the data are not shown there, but also we found PTSD cases are more likely to be a smoker or single status, and also more likely to have lower socioeconomic status, and more likely to report depression, anxiety symptoms and the assessment. And similar patterns have been found for Sanju Life, which is not shown here as well. And all those slides were unpublished data, and we appreciate if you do not copy or distribute those findings. And so this is a Manhattan plot that we found. There are one marker sort of reached the genome-wide significant association associated with the PTSD cases in CCSS. And also we found another 9 markers where risk loci sort of passed the marginal significance level. And we tried to replicate those findings in the Sanju Life. So for the top locus that from chromosome 10, that we have replicated the findings in Sanju Life, at a nominal significance level, but the estimates are very similar. And we did a meta-analysis we even get like more significant results after that. And in model 2, we try to adjust it for the cancer characteristics like age and cancer diagnosis or cancer type and cancer treatment, as well as other socioeconomic status. The associations are rather stable. And another locus, which was marginal significant in CCSS, and were kind of also replicated in central life. And the meta-analysis showed genome-wide significance, but also, I mean, the associations are very similar between CCS and central life, but we didn't have a very strong associations after adjusting for cancer treatment, etc. And the other 8 markers were not replicated in central life. So the other analysis, we focused on those 2 top or replicated markers that we did a number of sensitivity analysis. The first is trying to have more strict criteria on the PTSD cases classification. So we restricted our cases to those without depression and anxiety, and we have seen very similar associations, so that the depression and anxiety are not the driving forces here. And also we restricted to those cases with very significant distress or functional impairment, sort of higher up the case criteria. Also, we have seen similar associations. And we also did this analysis for the symptom scores, like total symptom score and also the sub-symptom scores, which all have very similar estimates of the associations, although the p-values were sort of not so strong in those sub-analyses. And also very interestingly, we compared our results with the other PTSD GWAS in the general population or the veteran populations. So for example, like those 2 markers were identified in our CCSS samples, but none of the associations were replicated in the PGC or IPSC GWAS. The P for heterogeneity is very significant here. But interestingly, for the other the markers identified in the PGC GWAS, we have observed like very similar associations in CCSS, but also, I mean, the p-values are not so strong, but it's not heterogeneous from the associations in PGC GWAS. And also for the IPSC GWAS, this marker is also have very similar associations showing here. So that's very interesting findings that sort of saying what we found in our cancer populations are not observed in the veteran or other general populations. And those markers found in those big consortia GWAS sort of we have observed very similar associations in our samples. And we also did a cross-trait associations using the polygenic risk score analysis. For example, like we use the PGC PTSD GWAS to calculate using their top genetic markers to calculate a genetic sort of risk score, which highlights the genetic predisposition for the PTSD in general population. And we analyze this score with the PTSD phenotype in our CCSS samples. So basically we found that the PRS for PTSD, PGC PTSD and major depression, anxiety, and ADHD are positively associated with PTSD in our CCSS sample. And also the PRS for subjective well-being was inversely associated with PTSD in CCSS sample. And this sort of suggesting that there are a lot of mental traits might share the genetic markers with the PTSD in cancer, so childhood cancer survivors. And we also did functional annotations via FUMA or MAGMA, then showing the top risk locus from chromosomal was associated with the gene expression of ECHS1 gene, which is associated with the mitochondrial function. And the locus on chromosome 6 is mapped to SASH1 gene, which is associated with the inflammation process. And we also did pathway analysis, enriched 16 very significant pathways associated with our phenotypes that lending further support to the involvement of energy production process and immune regulation pathways. Of course, we have several limitations in our study. For example, like we identified our PTSD cases based on DSM-4 criteria, which may not reflect it on the current DSM-5 criteria. But we also did several like sensitivity analysis to have more strict case definitions that the association remain very robust. And also the PTSD symptoms was like one-time measure, like 24 years after the cancer diagnosis or treatment. We basically captured the most severe or persistent like PTSD cases like years after cancer, but we may not capture those who are transient or they sort of have been the symptoms have been relieved like years now. And also in our controls, we might have some participants have milder or subspecial PTSD symptoms that would lead to attenuated associations. And our replication cohort Central Life includes PTSD after other traumas, in addition to the cancer experiences. But we have observed very similar prevalence of the PTSD in both cohorts. In conclusion, our findings suggest that the genetic susceptibility may contribute to the PTSD development among childhood cancer survivors, and there is clear evidence on the shared genetics with other trauma-related PTSD when we're comparing the results of ours to the other PTSD GWAS results. However, the top variants in our what we have found in our sample might which is not specifically associated with PTSD after childhood cancer, may highlight some heterogeneity of underlying biology for future research. And with that, I would like to acknowledge my mentor, Christopher Recolidis, Uneva Dimashtotti, and Karistan Koinen, and also CCSS leadership and colleagues and co-authors from CCSS and Central Life. Thank you, and the founders. Thank you.